Hello everyone, this is Frank Todaro, and I'll be presenting Dupret et al. The name of the article is Spatial Learning Depends on Both the Addition and Removal of New Hippocampal Neurons. I'd like to begin by explaining some definitions that are vital to this article, specifically neurogenesis. Neurogenesis is the process by which neurons are generated from neural stem cells and progenitor cells. Through precise genetic mechanisms of cell fate determination, many different varieties of excitatory and inhibitory neurons are generated from different kinds. Apoptosis is the death of cells that occurs as a normal and controlled part of the organism's growth or development. Developmental neurogenesis is most active during a baby's brain being formed, and it does continue throughout adulthood, but new neurons are generated at a slow pace, of course. And it's believed that neurogenesis is involved with learning and memory. Also, we must understand the synaptic pruning process that occurs in the brain. The brain gets rid of neurons and connections that it doesn't use or are weak in order to strengthen the neurons that are and the synaptic connections that are important. It's a way for the brain to maximize its efficiency. In mammals, adult neurogenesis occurs in the dentate gyrus in the hippocampal formation. The authors explained that the rate of neurogenesis is positively correlated to hippocampal-mediated learning abilities. Next, conditions that increase memory performance enhance neurogenesis, whereas conditions that decrease neurogenesis impaired learning. Lastly, spatial learning has been shown to increase both the survival of newborn neurons and cell proliferation. The authors cited that in rats with the lowest number of newly born cells had the best memory performances, indicating that learning and not training decreased the number of adult-born cells when they learned to navigate in a water maze. Hence, that increases and decreases in the number of newborn neurons are both correlated with learning. Dupret et al. Hypothesized that spatial learning is accompanied by events that are similar to that selective stabilization process observed during development. During brain development, many more neurons are produced than actually needed, as I mentioned earlier about what I saw synaptic pruning. And their study found that learning had three effects on neurogenesis. Learning promoted the survival of relatively mature neurons, induced the death of more immature neurons, and finally stimulated cell proliferation. Cell death, or apoptosis, seemed to be a pivotal event in this cascading event because blocking learning-induced apoptosis inhibits the other two cellular events and impaired memory abilities. Their results indicated that spatial learning involved a cascade of events similar to selective stabilization process by which neuronal networks were sculpted by adding and removing specific populations of cells as a function of their maturity and functional relevance. Briefly, the methods and procedure were a water maze training, as you can see in the photo. 3-month-old male Sprague Dolly rats were tested, and several animal subjects also. The researchers conducted several different experiments, and some of their procedures involving the animal subjects included cerebroventricular surgery, which was used to inject the animals and to get past the blood-brain barrier. Also, immunohistochemistry was used, and the authors explained that some animals were sacrificed. I take that to mean some animals passed away. Again, as long as all ethics were met, that's okay. Okay, the results. In the first experiment, the animals trained in the water maze, and there was 3 groups. The learning group L, they were compared to 2 other groups. Yoked group Y, those are the animals that were submitted to the same procedures as the control group, except that they were also exposed to the water maze for a time period equivalent to that of the learning group, but in the absence of the escape platform. So at the end of the training, the cell that was assessed in the animal subjects, the results found that spatial learning induced apoptic cell death in the DG. Learning-induced apoptosis was associated with a decrease in the number of the newly born cells that were produced during the early phase of the training. And, the researchers argued that cell death was specific to spatial learning. They found an increase in apoptic cell death only in the learning group. The visible platform, the control, and the yoke groups didn't differ. I just wanted to show a visual representation of apoptic, newborn, and proliferative cells through imaging. For the second experiment, Dupret et al. Determined whether learning a task that didn't require the hippocampus also increased cell death. They included a 4th group of animals trained to find the visible platform here. In short, it was found that the cell death was specific to spatial learning. Thus, they found an increase in apoptic cell death only in the learning group, whereas the visible platform, control, and yoke groups did not differ at all. The third experiment, the researchers examined whether a particular phase of the learning process was responsible for the induction of apoptosis, and you can see through matches 3 through 6 here. So it was determined that there was 2 phases that could be distinguished: an early phase during which the performance improved rapidly, and a late phase during which the performance stabilized and reached a symptotic level. Also, the authors note that since the 2 previous experiments for the control of the yoked animals didn't differ, only the control group was used for this and the subsequent experiment. The results found that only this asymptotic phase of learning induced cell death, and an increase in cell death was seen starting from the 4th day of training. The authors concluded that the results of their experiments reported that spatial learning promoted the survival of adult-born neurons that were relatively more mature and induced the death of the cells that were more immature, and finally stimulated proliferation of precursors. Regarding blocking learning-induced cell death, it showed an interdependency of those events and their involvement. By blocking learning-induced apoptosis, it inhibited cell survival and cell proliferation, and it impaired memory abilities different from learning. Their results indicated that spatial learning could involve a cascade of events similar to the selective process by which neuronal networks were sculpted by adding and removing specific populations of cells as a function of their maturity and functional relevance. There was a difference between mammal and birds concerning the relationships involving learning, neurogenesis, and apoptosis. So I think this quote sums it up, that in birds, apoptosis serves as a substitution of old learning for new, whereas in mammals, apoptosis seems to allow the efficient adding up of new information. So finally, the author's results showed that spatial learning involved a mechanism similar to the selective stabilization process observed during the brain development, in which the production of new neurons was followed by an active selection of some and removal of others. Spatial learning was not only based upon the additive processes that ranged from synaptic strengthening to the formation of the new synapses and new neurons, but was based upon regressive phenomena, specifically neuronal apoptosis. The epigenetic specification of the networks by removal of neurons in the adult brains provided evidence of additional mechanism contributing to the establishment of memory formation in animals. So there is a difference between the learning and memory. In closing, I thought this was a very well done journal and study. I thought the researchers were very thorough. They conducted several experiments. They used different groups to test. They did use animal subjects, and some people are against it, especially when some animals pass away. The authors did admit that additional investigations would be required to determine whether the adult-born neurons exerted a functional role in memory formation before or after reaching complete maturity. Curiously, I don't know if this is normal. I am only bringing it up because I've rarely seen this. They put materials and methods section last, following the conclusion. The authors admitted they had a discrepancy between the first experiment, but not the third. They explained that possibly it was because of the age of the newly born neurons that was studied in the two experiments. The first experiment, batch 1, had a decrease in newborn neurons. They were 5 and 8 days old at the time of the sacrifice, which I'm assuming in the third experiment, batch 3 to 6, the cells were less than 5 days old. Overall, and my final point, I thought it was interesting that the researchers blocked the apoptosis, and it showed that it inhibits the cell survival and cell proliferation, and that impaired memory abilities. And that learning-induced apoptosis was a very specific phenomenon. And I guess the most striking of their study was that the last interesting finding was that the learning-induced increase in cell death was correlated with spatial abilities. That is, rats with the highest number of dying cells had the best memory performance, and the authors asserted that the observation confirmed that spatial learning, and not the training, physical activity, or the stress, increases apoptosis. Okay, that'll be it. Thank you.